Hello and welcome. My name is Matthew J. Greer, and today I'm going to be giving a presentation on the molecular basis for skeletal muscle contraction. We're going to talk about what skeletal muscles are, why they contract, what that does for us. But we're going to be looking at, on the base level, what happens at the cellular level, at the microscopic level. This is prepared for Bio 515, Concepts of Human Physiology, through Grand Canyon University. My professor is Dr. John Tassone. And let's jump in. So the basics, some real quick stuff about muscles. Muscle cells are responsible for movement in the human body, and they accomplish this by contracting, by shortening. We have thin filaments that are going to pull these Z lines here on the outside. And what happens is as those get pulled in, they contract and they shorten so that the space in the middle here is taken away. This is pulled in together. And this sarcomere, this kind of functional unit in muscle cells, is going to contract. And this is called the sliding filament mechanism, because these filaments here are sliding in. So let's break that down a little further. We got to talk about actin and myosin. Actin's this one down here on the bottom, and myosin's this one here on the top. And a single skeletal muscle or fiber has many microfibrils. These stretch the entire length of that fiber, of that cell, and as they shorten and they contract and that sliding filament closes, that cell contracts, and if all is contracted in muscle, they cause contraction of the muscle. And it allows you to walk, to interact with your environment, to move things, manipulate objects, all kinds of good things. These have thick and thin filaments, these microfibrils, and a thick filament has mostly myosin, this one on the top, and the thin has mostly actin, this one on the bottom. Actin, each one of these little dots in here is an actin, and this little dark blue spot that I'm hovering my pointer over, those are spots where myosin will bind to these guys. We're going to talk about tropomyosin and the troponin complex in a little bit, but myosin has these two heads, and it has this tail that's kind of like braided going back. And these are very important. These two interact with each other to create that shortening. So there are some regulatory proteins to contract for muscles to actually do their job. That myosin with the heads from before has to bind to these little blue dots. But unless the cells want to and allow to that contraction, it can't happen. And the cell has these two proteins that are these regulatory proteins that protect these binding sites from being binded. The first one is tropomyosin, and it is this kind of pink filament looking one here on this one. And as you can see, that's going right over those active sites, and it kind of locks them, prevents that myosin from reaching up there and binding to those active sites. And then troponin, and this troponin complex is this pink kind of going back in there. They have 3 polypeptide unit of a protein. They have 1 polypeptide that bonds to actin, 1 that binds to tropomyosin, and 1 that binds to calcium. And without the calcium here in this top unit, this is locked. It's unable to do any kind of binding to myosin. With the addition of calcium ion, we now have some of these active sites open. We can now see some binding happening and actually have some contraction. So this contraction, this is called a power stroke. And this is when the tropomyosin and troponin are removed by that binding of those calciums. Now these myosin heads here can bind to these actin molecules. And as they do, they're going to walk up this actin, and this is what shortens and contracts that sliding filament. Each stroke here, as it's pulling it towards the center, is called a power stroke as you're moving towards the sarcomere. There are many power strokes in each muscle contraction. To get this signal started, we need to, like in nerves, we need to activate or kind of get this action potential propagated inside of these. Skeletal muscles are stimulated to contract by acetylcholine. This molecule is released by nerves from the central nervous system in the neuromuscular junction. And as that action potential spreads into the muscle cell, it is going to travel through the sarcolemma. And this is the plasma membrane of muscles. As that action travels through the sarcolemma, it's going to travel down this T-tubule, which is a transverse tubule because it kind of moves transversely. And as it comes down, it's going to go through here, and it's going to start moving towards this sarcoplasmic reticulum over here, this SR. So to get these muscle cells contracting and to get this action potential through, we have to release calcium ions. In the sarcoplasmic reticulum, this little structure over here on the right, we have lateral sacs that store calcium ions, Ca2+. To activate these guys, the action potential has to go to that dihydropyridine receptor on the T-tubule, which gets activated by depolarization. And in turn, these foot proteins that are connected here also get activated, and they release that calcium ion. We call these ryanodine receptors. And as they get activated, this calcium starts to flow into the cytosol. And as it flows into the cytosol, it is now available to free up that binding site of actin. It's able to bind to that tropomyosin and get that moved aside so that we can start that binding process. Now, ATP also has a role in muscle contraction. The myosin cross bridges, these myosins have two active sites. Myosins have two acting sites. The first is the actin binding site, and this is the one on the top that's going to bind to this actin molecule and create the cross bridge. And then we have the ATPase site. And this is where you can see this ATP molecule binding and getting split into ADP and an inorganic phosphate. After myosin binds to this actin, ADP and PI detach, and it leaves it ready to have a new ATP molecule bind. And when that new ATP molecule binds, that's when this cross bridge breaks, and the process is able to start over again, and that's able to shorten more. It's able to move to the next actin binding site and continue to bind its way and walk down this molecule. Now, we can get our muscles tight. We've talked about that, but how do we relax them? Because you have to be able to relax. Relaxation in your muscles is getting this calcium back out of the cytosol. With this calcium out here, we can't use that troponin-tropomyosin complex to block those binding sites so that muscle is able to contract and that myosin is able to bind to that actin. So the sarcoplasmic endoplasmic reticulum, Ca2+ ATPase, or more commonly and easily called the circa pump, is kind of responsible for this. It is going to take the calcium from the cytosol, and it is going to concentrate it back into these lateral sacs in the SR. This allows the troponin-tropomyosin complex to slip back into place and block the attachment of myosin again. So now this muscle is allowed to relax and is no longer contracting. Rigor mortis is an example of what happens when this doesn't happen. Rigor mortis, also known as stillness of death. It sets in after death 3 to 4 hours, and it lasts until about 12 hours after death. It's very important for forensic science. It's used to determine time of death. It's used to determine, how long the body has been there. At this time, after the body is dead, this calcium leaks out of these lateral sacs. And now, this myosin is allowed to bind to actin because those binding sites have been moved across. The troponin-tropomyosin complex has been removed with this excess calcium. So these muscles are allowed to attach and contract. Now, because the body is no longer alive, we don't have any fresh ATP. So those muscles can't release. So that's why the body is stiff. That body is seized up into like a constant state of contraction. So having those calcium, that circa pump is very important. Without it working, without you being alive, that circa pump can't work. It can't release. It can't reuptake that calcium. And the muscles aren't allowed to relax. This only lasts about 12 hours because after 12 hours, those proteins and complexes start to degrade, and the muscle is finally allowed to relax again. Well, that's the conclusion of our discussion on the molecular basis for cellular contraction. I hope you have a better understanding on how a sarcomere is able to cause contraction on the molecular level. I hope you understand the involvement of actin and myosin, the troponin and the tropomyosin complex for preventing that myosin from binding to actin, the importance of calcium to release that complex and get that kind of primed for that myosin to attach and start the actual process of contraction. And I hope you also realize how important the circuit pump is in removing that calcium from the cytosol so that that muscle can relax. And relaxation of the muscles is a very important process. You kind of have to have that give and take. If you want to look into more information about this, the textbook from Sherwood has a lot of good information about skeletal muscle. And then if you want to look at more of the rigor mortis and more of the forensic stuff, which is very exciting, Shrestha, this methods of estimation of time, has a lot of good stuff, not just rigor mortis, but has some good stuff about rigor mortis as well. I hope you learned something new, and I hope you enjoyed this. Thank you.